Hi everyone, thank you for joining Data Cornell 2022. Welcome to BI Reporting Business Use Cases. And my name is Minoy, your host, and your co-host today, Courtney and Harinda. Please put your phone in the silent mode. This session is currently streaming via Zoom and will be recorded, and it will be available online within a few weeks. Today, our guest is Angela.
Yes. Hello, everyone.
And she will be talking about early cancer detection using higher order genome architecture and chromatin interactions. Angela is data enthusiast who seeks insights from biomedical and clinical data for disease interception and prevention. So patients could be treated with appropriate therapeutic strategies to control disease development in advance. After graduating from MS in bioinformatics, she worked as a UX researcher at Apple for several months. Currently, she is bioinformatics research associate at Sigmatic, a sequencing service company in Bay Area. Without further ado, I will hand this over to Angela. Thanks for being here.
Thank you, Mark. All right. Hello, everyone. Happy Saturday. And thank you for attending DataCon LA. And today I would like to present a topic on effective cancer detection using higher order genome architecture and chromatin interaction. So to start with the project, I actually have a conceptual question. Is how do higher order genomic structures and epigenetic regulations influence the chromatin interaction in normal and cancer cell lines? So for this project, I actually built a detection models and to differentiate different cell types using some biological information. So one of them is a high order genomic structure, and the other one is epigenetic regulation. And the model was built with an accuracy for classification at 80.90%. Now let me walk you through how I do it and how I get the answer for this question. So here is a general roadmap of what I go through today. I will start to introduce you some scientific background, and then I will also summarize the data pipeline that I process from the raw data set to the target data set, where I can use that to build the features and to visualize the relationship between different features. And then after that, I build a detection model and also quickly go through the evaluation performance for those models. All right. So to start building a model, I need some clinical information and scientific background. So those are essential to guide the whole project. So on the left side is a pathological slide for the cancer patient. So these are pathological slides for the normal lung tissue, and these are for the cancerous lung tissue. So in general context, as an early stage in diagnostic, cancer patients have to go through painful procedures such as tissue biopsy to help doctor determine whether they have cancer or whether in their body the cells are cancerous. So the top slide here is the normal lung tissue with less crowd cell population, which is usually much less crowd, mostly less cell populations, and lower staining power. And the bottom slide is the cancerous lung tissue, which is much dense cell populations with higher staining power here. So the procedure is costly, time-consuming, and painful for cancer patients. Therefore, my motivation for this project is to use the biological information to build a detection model to detect different cell lines. So let me introduce you to this biology map here. There are three domains in this map, which is genetics domain, genomic structure, and epigenetics domain. A lot of research projects have been going on with the genetic domain. So, but in this project, we focus mainly on the genomic structure and epigenetics. All right. So now let's step into the genomic structure. I will introduce you briefly about the background for the genetic structure to help you understand what I do for this project. So in this project, for the genomic structure, it's divided into primary order and higher order structure. Under primary order, there are chromatin and heterochromatin. And under higher order structure, they have the active and inactive compartments. All right. When you zoom into those compartments, you will see a little folding structure. It's called topology associated domain, and abbreviated as TAD, which is tag. All right, and it's also divided into the active tag and inactive tag. Active tag has a lot of genes, transcription factors, and show early replication. And inactive tag has less gene and show later replication. Now let's work into the specific structure of the tag. Yeah. Okay. So now imagine that we have a string of DNA. It's a long string, right? And when in the string, there's a two card on both sides. That is the CT and CN anchor. And here is a cohesive ring. This is the string of DNA or DNA sequence. And then how it looks, it just the ring just go through, the string just pass through the ring here. And then until it hits the CTCN anchor. So most of the time, it should be around this structure, but sometimes the cohesive ring can stop in the middle. But that case is not the case that we will talk in this project. So go a little bit more into the structure of this. So if you look on this picture here, it's basically it also resembles the structure. So for the tag structure, it's made up of DNA sequence with the CTCN anchor and a cohesive ring. So here, CTCN anchor is a triple CTC5 vector, and this is also a highly conserved zinc protein, and it's also an insulation, an important insulator to control the gene expression. In this case, it is the regulator of chromatin organization. Cohesive ring is basically made up of protein complex, which is RAD21 and MC, which is, we don't need to go deep into that. All right. So what is this section? It's a DNA sequence. So in this section, they have some important region along this sequence. The purpose of the tag structure is to bring anything farther away come closer instead. So for example, I have enhancer on the side, and because I have enhancer on the side, right, and promoter on the side. This with the loop extrusion create this tag structure. It brings the enhancer and promoter closer in space to control the gene activity. Now, if in there are multiple enhancer occur, it will create super enhancer. And how the super enhancer works is very fascinating, and I will briefly go through it the next slide to show you. All right. So this next slide we step into epigenetics. That is where something happens to control the enhancer and super enhancer. So we have something called we have multiple different types of histone markers. And here in this project, I only use two types. So one type is acetylated histone 3 lysine 27. And this is the marker of the activator. When this activator present next to the enhancer, they turn on the enhancer. And this enhancer will be promoter to turn on the gene expression. So you see the orange. If there are multiple activators occur, it signifies the region of the super enhancer. So when the super enhancer occurs, it's actually turn on the genes, but it's even turn on even much more than normal level. So that's how, like, for example, cancer, right? So in cancer, abnormal level of gene expression. And the second type of histone marker I use in this project is trimethylation of histone 3 less than 27. And this is the marker of the repressor. When this repressor present, it turns off, it makes the cancer become inactive and it turns off the gene expression. Okay. All right, so how do we put both chromatin structure and epigenetic regulation in one space, and how to make sense of it, its contribution to cancer pathway? All right, earlier I mentioned about the chromatin structure, making the loop bring everything closer in space so that it can control the gene activity. And for the epigenetic regulation, which is the activators, right? So in this case, we have a high intensity of this activator, which is the acidolated histone 3 less than 27. With this high intensity, it signifies this is a super enhancer in the colon cancer and breast cancer cohorts compared to the normal cell. Like colon cell, normal colon cell, and normal mammary epithelial cells, which you don't see a lot of very high intensity of this area. All right. And then how these two put together into the cancer pathway. So increase in the super enhancer on the oncogenic and decrease in super enhancer on the tumor suppressor gene. And both have significant effects on the cancer pathway, on a longer cancer stage development. So it can make the cancer cell to proliferate, grow more, or it can make the cancer cell to start to migrate to other side body and then invade those regions of the body. And in some cases, those also make the cancer cell resist to certain chemotherapy. Okay, so the next step, how I put all of those scientific information together to build a pipeline. And then generate a contact matrix to show the dynamic folding of the genomic structure and how they're different in between different cell lines. All right, so the 4 cell lines I use here, because this is my master project. So the 4 cell lines I use here is basically the cell line that I published. And a lot of people use the cell line for the research purpose. So in this case, I use the lung fibroblast and lymphoblast for the 2 normal cell lines. And I use the lung adenocarcinoma and leukemia for cancer cells. Next. All right. So to obtain the contact matrix or the dynamic folding, how do we actually see what the variation between the dynamic structure in different cell lines? We use something called in situ HiC. This experiment is very fascinating. Generally, it starts with cutting DNA with transcription enzyme, and then it's ligate with the biotin, and after that, it wash away the biotin and perform the sequencing. So after the sequencing, we obtain the genetic information and also other genomic structure for the pipeline. Can we move to the next one? If you want to know more about in situ HiC, feel free to meet with me at the end of this presentation. All right. So the data pipeline that I have here is very complex. I'm trying to put it in the way here, like this way, to just make it easier for everybody to see. All right. So I start with the raw data. So the raw data that come from the sequencing is the fasting data. All right. So this is the fasting data. Have you heard about the Illumina? The Illumina sequencing? Yes. So it's so the result come out from the MySeq machine from Illumina is the fasting data. For other type of machine in Illumina is they generate BCL data type and then convert to fasting data by using some other tools. All right. So from fasting data, I converted it, I processed it multiple ways actually a lot of process happening here to get the target pricing data using Enthusiast. And then from this target pricing data, I use a point box to visualize the chromatin content matrix. This is how we do it. And I also use the Chung tool to obtain other quantitative features to classify different types of cells. In particular, in this case, there's all of them that we should disclose using time. All right. Next part. So after I get the target data with this Python data, I use a tool to create the content matrix.
Got it.
Just 1 second, battery's dying, so we try and get a charge on it now.
No, it is not coming.
It's not connecting again.
Connecting.
2 seconds. Oh, you should. Yes, sir.
Starting
Yeah,
sharing.
here we go.
Sorry.
Very sorry for the somehow I don't see the time.
Sure.
Oh, no, yeah. You make it put it down. So for the prompt,
This one, right?
no, that's first. This one. Okay, sure. Oh, okay, I can. Thank you. Thank you so much. Okay. All right. So remember I used the compound to generate a content matrix. So here is the animated version, the drawing version to make it easier to understand. So in this one is a part of the content matrix. If you look at this side, it's more like the external axis. And when they zoom into a particular regions along this external axis, they can see more squares. Okay, and in here, the square without the little dot on both sides, they are ordinary domain. And the square with the dot on both sides, they are the loop domains. So there are 2 types of domains for the context domains. So there's an ordinary domain and a loop domain for the context domain. Okay, so this is the actual version of the context domain. So here, you pay attention to the diagonal axis which has very high intensity, very dark, very intensity here. You don't see a lot of the activity going on here until you zoom it into the higher resolution. So here you can see more square become visible, right? And here we can continue to zoom down further until we don't see any more square overlap. So here see a lot of square overlapping here. And these squares here present the contact domains. Yeah, this one we have to move it. All right. Okay. Yeah, it's okay. So this graph have a lot of information going on, but just focus on the marker, like the read marker here. Say everywhere along our side. So here is also look like the previous one I showed earlier. This one showing subcompartment. Sorry. Why does it happen? And this is the diagonal axis. When I zoom into a particular location here, we see there's more square present. And when we zoom further into higher resolution, you can see more square become visible. And those squares showing the dynamic folding of the contact measures between different cell lines. And with the top here is lower resolution. When you go down further here is a higher resolution. So higher resolution, you can see more detail along that axis. How comes? Oh yeah, that's okay. So in this paper here, we have lung cancer cell line here, lung fibroblasts, B lymphocyte, and the last one is leukemia cell. So this is cancer cell line, normal, yes. Right. So in between the lung cancer and lung fibroblast cells, you can see some dynamic folding difference between these two cell lines. So here, if you look at these particular chromosome locations, there are more folding activity, more intensity of the squares, more looping activity after within the cancer cell, within the lung cancer versus the lung fibroblast. And when you look at the B lymphocyte leukemia cell line, they're not directly related to each other, but for the visualization purpose, I want to show here, which is in this case, at this chromosome location. And this one, they have very similar looping activity. And here, you kind of see that they're more looping activity, more busy in this area of the cancer cell line versus the normal cell lines. So earlier for the chromatin contact matrix, we can zoom into a particular very small region of the chromosome to see the different folding between different cell types. And now I go into the feature engineering and do some visualization to see the relationship between different features in the larger scale. Okay. So here is a flow chart to guide by feature building. So I do different features and using all of this information to create a tabular data format. And here's some visualization based on that feature. Okay. So using the feature coming out from that flow chart, the data engineering, I try to visualize the relationship between the chromatin state distribution in the presence of activator. So for the chromatin state, there are like multiple subcategories with a strong enhancer, active promoter, positive super enhancer, and negative super enhancer. So in this case, what we can see here is there are high number of strong enhancer, super enhancer, and active promoters in the presence of this activator. That's, in other words, the activate chromatin mark is positively correlated with the enhancer and the super enhancers. Oh, sorry. Yes, that's the one that I want to talk about. I just finished that. Okay, sorry. Okay, so for this bottom part here, it's showing different boundary strength distribution of different cell lines in the presence of this activator. So in this case, boundary strength is defined by some of isolation score, and also is the mark. It also shows how strong is the PEG boundary in terms of gene expression. No. Sorry, is that it? Okay. Yeah, so in this case that we can see the high density of the strong boundary strength. I'm sorry. For this case, the boundary strength is divided into different levels. The boundary strength 4 or boundary 4 or 3 is the boundary strength with is the highest boundary strength and is signified for the strong protection. And those are boundary strength 1 and 2 is the weakest boundary. So in this case here, we can see that the high density of the strong boundary, which is 3 and 4, is correspond well to the high number of enhancer and promoters in the presence of this activator. Yeah. So the executive summary here is that with that condition where they corresponding well between the high number of enhancer promoter and the strong boundary, this simulate a condition that the super enhancer and the strong enhancer tend to localize and co-duplicate at the strong boundary to be protected from deletion. So using the same flow with the previous slide, in this case, I put it in the context of request. So now let's focus on the normal cell type. So here, these are the 2 normal cell lines that we have. So there are some negative super enhancer in the presence of different reversal. And this indicates a potential decrease in gene expression. And when you look at the boundary strength here, it's boundary 1 and 2, which is the weak boundary. And this weak boundary corresponding well to the negative super enhancer and the repressor. Now let's shift our focus to cancer cell lines. So in these cancer cell lines, the low density of negative super enhancer and weak boundary occur in the presence of this repressor. And in this case, this indicates that the cancer cell prefer the strong boundary over the weak boundary to protect its super enhancer to be big, so that it can maintain the abnormal level of gene expression. Yeah, I mean, that's summary. All right. So one of the main purpose of this project is to build a model, a vegetation model to differentiate different types of cell lines. So so in this project, I tried 3 different machine learning algorithms here. So I will briefly go through the definition of each and then we move on to evaluation components. So for random forest, it is an sample learning, which allows learner to enrolling with some sample, right? And seeing generalized data by randomly combining like different subset of features on the vision tree is learn a variety of things to reduce the overfitting. For XGBoost, it is an extreme gradient boosting method. It's a large sequential tree rowing with weight sample. And XGBoost has a regularization parameter, learning rate is shrinkage for beta boost with the adaptive boosting, and the columns of sampling for random forest to reduce the variance in the noisy data set and to reduce overfitting as well as increase generalization. So TAPNet here is a neural network, and it's employed the block of feature, attentive transformer, and mask containing fully connected layer and normalization techniques. And these components allow us to select feature importance to improve the accuracy for classification. So in this project, with the data I have, the XGBoost gives the best performance at 18.90%, and then followed by random forest and TAPNet. Because these 2 models have better performance than the TAPNet, so I will focus on the evaluation performance for these 2 in the upcoming slide. Okay. So after performing random forest algorithm and also Chung